Mass spectrometry is used to analyze protein molecules, and more specifically, can be used to study post-translational modification of proteins. Before we go into detail about mass spectrometry, it's important that we understand what it's analyzing. So let's start from the beginning. What is a protein? Well, simply put, a protein molecule is a string of amino acids held together by polypeptide bonds. You can think of this like a bunch of beads looped together on a single thread. This string then goes through a series of processes that fold and combine the proteins to end in a final functional form. However, even in this final form, many proteins still require post-translational modifications to actually function within a cell. So where does the term translational come from? Well, this has to do with how a protein is made. It all starts with DNA, which then goes through the process of transcription, which results in RNA. You can think of transcribing as copying the pages of a book onto a separate sheet of paper. mRNA can then be used as a template in the process of translation, which will result in a basic protein molecule. Think of this like translating the English text you copy down into Spanish or some other language. Thus, post-translational modification refers to modifications made to a protein molecule after this process of translation. Now, there are many different types of post-translational modifications, but one that I would like to highlight is phosphorylation. Phosphorylation is the process of adding or removing a phosphate group to a protein molecule. In doing so, the protein can be activated or deactivated, almost like a light switch. Thus, a single protein can now perform two different functions. This leads us to one of the most important results of post-translational modification, which is increased proteomic complexity. We start out with only a set number of genes. Through the process of transcription and translation, the cell can select and activate these genes in a way that can code for over 4 times the number of proteins. However, with post-translational modifications, we now have over 1 million protein species. That's over 40 times our original genes. Now, how is this possible? Well, recall the example of phosphorylation I just shared. By being phosphorylated, this single protein has 2 subspecies, on and off. And that's only with phosphorylation. The same protein can undergo many other types of post-translational modifications, creating even more species or forms of this protein. Not only does this make the cell extremely efficient, it also makes it possible so a set number of proteins can now have the capability to carry out exceptionally complex cellular processes through its different forms. As such, understanding these post-translational modifications is beneficial because it increases our understanding of important biochemical pathways, including the pathogenesis of diseases. Take, for example, HER2-positive breast cancer. Researchers have found that when cells have a greater amount of these activated HER2 receptors, seen here, this is associated with poor prognosis in HER2-positive breast cancer patients. Through the study of post-translational modification, they found that HER2 receptors are activated by phosphorylation, just like our previous example. By studying post-translational modifications, we can now use phosphorylated HER2 receptors as a biomarker for breast cancer. This is just one of many examples of how the study of post-translational modifications can have important real-world applications. Thank you all for watching this video. I hope you enjoyed it and now have a better understanding of post-translational modification. This video was created by me, Gabriella Goodwin, as a member of Dr. Ryu's computational lab group, and it was funded by the National Science Foundation. Finally, I'd like to end by thanking Dr. Ryu, as well as the members of the PTM Educational Application Group and the remaining members of the Ryu Computational Lab Group, without whom this research would not be possible. Thank you.